The CHIC project was aimed to see how genome editing could be of benefit to consumers in a small crop. And for that we choose chicory, which is a relatively small crop, and with small I mean grown on a little areas.
At the start of the project, very little was known about chicory in the lab. So there were some protocols available, some publications, literature. But specifically for genome editing, we had to set up this in chicory itself. And so I think the main challenge was the uncertainty that we had at the start of the project. Would this be possible in chicory? And would it work effectively? But I think we overcame them quite well, and we ended up with a very efficient method to do what we would like to do.
For using these new plant breeding techniques, you really need to have a lot of information about the genome of the plant, so the DNA of the plant, in order to really target the genes that you want to target, but maybe more important also to analyze what you did afterwards. And that we thought in the beginning that we would have a perfect information, genomic information, but it turned out that it was not really good, so we had to redo this.
We are really dependent on getting the data, and for us it would be really much more easier if there would be a large-scale deployment included in the project, but this was not aimed for because it was a research project. So our assessments are based on model data. There we get some good data to analyze, but in the next step it would be very good to also have a large-scale deployment, field trials, for example, of the chicory roots, and then to also redo this assessment and to compare the results also, yeah.
We more or less started from scratch on this project. So we had to generate a lot of background knowledge, like new genome sequence, new transcriptome sequence, to really allow us to, move forward in the project.
With plants, we cannot, certain steps we cannot speed up. And then related to this kind of project concept, so sometimes the tasks are dependent on the other tasks, so we needed to adjust the time scaling in that aspect. But on the other hand, we could overcome these limitations in a sense that, for example, within certain methods we could develop and implement the methods already with some other similar type of material, and then when the actual material is ready we can proceed very fast.
So we had a little bit delay in the assessment, but due to the prolonged project runtime now, it's not a problem anymore. And another difficulty that we have faced is that in Europe, genome editing is still seen as a GMO, which makes comparable to classical GMOs, so it's nothing new. So we had to focus on a potential future risk assessment, which could be based on a different regulatory regime.
In mid-2018, the ruling of the European Court of Justice clarified that all types of genome-edited plants are being regulated in the same way as GMOs. That was a big surprise, was not expected by our project consortium. It was not expected by the majority of observers of the process.
Well, the main challenges were to try to translate one's CHIC into an easy to understand language, okay, for school students, for citizens, for artists, but at the end we made it, okay, it was a lot of work. Sometimes I thought we were not going to do it, but finally we did it, yeah.
These are people from many different disciplines. We have social scientists, we have medical scientists, we have biologists, we have communicators, we have stakeholders. And actually, so I thought that might be difficult to have them all aligned and all have going for the same objective, but actually that was not a problem at all. So it turned out that it was much more easy than I thought.